Hi, this is Mr. McLean. In this section, I want to take a look at how the light reactions work specifically. I already have two photosynthesis videos that I've created and posted. The first one is more of a general overview. The second video emphasizes the characteristics of light, taking a look at the absorption and action spectrum and how that relates to the light. So in this particular video, what we're going to do is we're going to follow the flow of electrons in the light reactions. And when you look at this particular diagram that's in our notes as well as our textbook, we can see that this is depicting what we would consider the linear flow of electrons, in the sense that it's eventually going to deliver those electrons in the form of NADPH. So this would probably be a more complete image right here. We're going to see the flow of electrons coming in from the source of water and traveling to the Calvin cycle, which is our sugar-producing process that occurs in the stroma, coming in the form of NADPH as well as the ATP. So we looked at a diagram in class, and I'm just going to go over that very quickly with you to make sure that everybody understands exactly what's happening with this. So just as a quick review, these two columns are representing our photosystems. So I'm going to lay that down there for a second, and I'm actually going to see if I can modify that. Okay. Okay, so we hid my picture there. All right, so in the first column over here, we have what's referred to as photosystem number 2. This one over here is photosystem number 1. And what's happening is we have simultaneous photons striking each of those light reaction centers, whether it's in photosystem 2 or photosystem 1. As the photons are striking the various accessory pigments, they're exciting the electrons in those pigments. And at the center of both photosystems, we have chlorophyll A. All right. Now, chlorophyll A in the photosystem 2 is referred to as P680 reaction center. So that's going to be most functional at the 680 nanometers. This one over here is P700, so it's slightly different as far as where it's most effective at driving photosynthesis. All right, so what happens is as the light strikes the reaction center, the electrons in the pigment chlorophyll A become excited. And what they do is they get captured by a primary electron acceptor. And a primary electron acceptor is a molecule that is going to be an oxidizer. So it's going to catch that electron, and instead of allowing it to just drop back down like we would see in fluorescence, what it's going to do is it's going to transport that into an electron transport chain. Now, for the sake of the AP test and for the sake of my class, you don't need to know the specific components of the electron transport chain. But what we do know is that that energy is coming off in a controlled fashion. And that exergonic release of energy is coupled with a proton pump that's going to push hydrogen ions across the membrane. And what it's going to do is it's going to build up a hydrogen gradient inside the thylakoid space. Okay, so this is my electron transport chain. We'll just say ETC. Simultaneously, we have the excitation of electrons that are happening right here at P700. Now, one thing to keep in mind, once this loses an electron, it now becomes P700 plus. Okay, so it's short an electron, which means that it has a oxidizing characteristic that it wants to pull that electron going down. Same thing over here. Once this has lost its electron, it has become P680 plus. And it is also pulling electrons. Where it's pulling its electrons from is water. Okay, so water is the source of our electron for this particular photosystem. When the water is entered in there, it undergoes an enzymatically catalyzed reaction. we get some oxygen coming off, we get hydrogen, and we get electrons. All right, so back over here, you have two processes that are simultaneously occurring. Excitation on the photosystem 2, excitation in photosystem number 1. Once again, you got an electron that is going to be caught by a primary electron acceptor, and the primary electron acceptor is going to drop the electrons into another chain. Now, I do want to point out this is Ferredoxin. And Ferredoxin has a unique characteristic that I'm going to show you here in 1 second. And it delivers its electrons down to NADP reductase, which is an enzyme. So the idea is that we're going to take NADP and we're going to reduce it into NADPH. Okay, and like I said, this is going to the Calvin cycle. And over here, the hydrogen gradient was going to make chemiosmosis possible. So as that chemiosmosis takes place, we are able to couple the downward flow of hydrogen through ATP synthase to produce ATP. So we have ATP and we have NADPH, which are going to go to our Calvin cycle. So if we just kind of review this one last time with this here, our flow of electrons have us coming in. We have, like I said, we have our situation where we're going to start in with the hydrogen and the electrons being donated from water. As the water is split and the electrons and the hydrogen are made available, what we'll notice is we'll see a flow of those electrons to this P680 plus. The reason why it's P680 plus is that it had previously lost its electrons due to excitation. It's going to come down the chain of the electron transport chain. We simultaneously have a second photosystem over here. We refer to that as PS1. Now, one of the things that I wanted to make sure that I added is over here on this side, we have what's referred to as the possibility of cyclic electron flow. In fact, that doesn't show up too well. So let me change that a little bit. Let's go with something that's going to be a little bit different. Let's see if I can see if green will work for me. Okay, so if you have cyclic, well, it's not working. If you have cyclic flow, what's actually happening is that Ferredoxin, instead of delivering it over here to the NADP reductase, like we see in this overall linear position, cyclic is only going to focus in on this portion right here. And Ferredoxin, instead of delivering it to NADP, it actually drops it into the cytochrome complexes of the first electron transport chain, and that produces ATP only with no production of NADPH. So that is a evolutionary relic, if you want to think of it that way. It is a reaction that is not it's not really seen in a lot of our common photosynthetic organisms. We find it more in the purple bacteria and the sulfur-reducing bacteria. Okay, so hopefully that helps clear up the light reactions, and hopefully you understand how that ties in with photosynthesis as a whole.